Sure, I have a million choices.
Here's what I want you to do today. This is to review. So, see how I put a number 1 here?
Yep.
And then, so 2, just show you them. 3, 4, 5, 6, and 7.
Yep.
So just you're going to put a number down the margin. Here's what I'd like you to do. Write the name of the molecule.
Okay.
What act is it in? Name the act.
Okay.
And name the type of reactions that are going on.
Okay.
Okay? So I'm going to give you maybe 3.5 minutes to do this in total, right? So this is just practice. If you are totally stuck and nothing's coming into your great brain, just move on to the next one, right? And then obviously we'll take these up in a couple of minutes. We'll just start with you, then Victoria. If you don't know the answer, you just say pass, right? And then Lauren, James, etc. So, what do you have for number 1, young lady? Pep, good. What act are we in? Not a girl. And what's the name of the reaction? It is, but it's a specific type of phosphorylation. Substrate. Excellent. All right, Victoria. Can you name the carbon molecule? Great. Do you remember how you remember that?
Because it has, I think it's great. It has a lot of
electrons. So you're thinking that's energy. So the great means there's more energy in this molecule than 3 Pg, right? That's that G connection that you got. Yeah. Okay, correct. G3P. What act are we in? Good. And what's the name of the reaction that's about to occur? And reduction. And we put those two together to say that this is a redox reaction. Good. Lauren, carbon molecule?
It's an acetyl group.
Acetyl group. Act?
Pyruvate oxidation.
Type of reaction?
Redox.
Excellent. James. Name of the molecule?
Malate?
Excellent. Malate. What act are we in?
Krebs cycle.
And what type of reaction is going to take place?
A redox reaction.
Good. One trick question here, not trick, but extra. What is different here about this redox reaction compared to with NAD plus?
Yes. The fat has takes on 2 hydrogen instead of just taking on 2 electrons and putting off 1 proton.
Excellent. Joanna, you've got most action. What's the name of the 5-carbon molecule? Atta girl, ketoglutari. What type of reaction is going to happen between the carbon molecule and the coenzyme? And what's the other kind of reaction that's going to happen? Good, and what kind again? You indicated over here.
Substrate.
Good. What's the substrate in this reaction? Okay, Victoria, do you know what the substrate is here?
No.
Okay, Lauren?
It's the PEP.
Yeah, the PEP. So just go over this in your mind again. The substrate is what? The substrate is the molecule that fits into the what?
The active site.
The active site of what? The enzyme. That's the substrate. Okay, so we have substrate level, a substrate level phosphorylation reaction is going to happen at this point in the Krebs cycle. Lauren, do you remember what molecule will become the substrate?
G. TP?
Exactly, because it's not present in that picture. Good. James, 6 carbon here. 6 carbon. Picture 6, 4 carbon.
It's oxaloacetate.
Good. So what reaction just occurred?
Redox.
Excellent. Victoria, were you here yesterday?
Yeah.
Okay. So, we don't have any carbon molecules here. What's the name of this protein? It's the first protein in the electron transport chain. Yeah, you're. That's right.
I can see it in my head. I don't
So
know
look,
what to say.
think about the reaction that we saw yesterday that we'll go over again today. Look, NADH loses a hydrogen. This is called NADH dehydrogenase. Right? Okay. What reaction occurred here? What type of reaction? Substrate phosphorylation or redox? Yeah, but your confidence level's not high in that, is it?
No.
Okay.
No, but it makes sense now that I
Yeah,
say it.
so the redox is when electrons
Yeah.
are being transferred from one molecule to another, it's a redox reaction.
Yeah.
Okay, excellent. Questions?
About number 2.
Yeah, I just want to take these numbers down. Yes, let's go.
Because there's the phosphate that's going to be put on to make BPG, is that also a substrate level phosphorylation?
Excellent. No.
No? Okay.
Because this is not a substrate.
Oh, okay.
It's not being right?
Yeah.
That phosphate is just being transferred to carbon 1.
Okay.
Right? Excellent question. Why is glycolysis anaerobic? Why do we classify it as an anaerobic set of reactions?
Because it doesn't use, it's either glucose or energy.
Okay, well, was glucose at the beginning of this story?
Yeah.
Yeah.
Doesn't use oxygen.
Oh,
Yeah, there's no
that's
oxygen present, correct?
Yeah.
Okay, let's look at where we left off yesterday. Right here. I believe we're set, ready to go. So do we have any questions? So looking at the first reaction, we did quite a bit of work there. Have you read over your notes? Let's do a quick summary, shall we? Okay, so look. So we followed electrons on what molecule, Victoria? Glucose. She says with a ton of confidence. Yes. This is the story we're following. Joanna, is there energy in the glucose molecule? What form is that energy in? It's in electrons. So, Lauren, is there energy in this water?
Yes.
Is there more energy in it now if I pour it compared to if I pour it here?
Yes.
So the mitochondria, and this is where you were getting frustrated yesterday, right? Because only after all that work, only 1 proton. And you're like, you gotta be kidding me, right? This is the 1 proton being pumped up here. We're gonna see more protons get pumped up. When the energy in this glucose molecule is transferred to these various coenzymes during glycolysis, pyruvate oxidation, Krebs cycle, right? This amazing set of proteins now, called the what? What are these set of proteins called?
Electron transfer.
Yeah, these amazing set of proteins are going to use the energy that was in glucose to move protons from where? From where?
The matrix.
To the
Inter? Is it inter or inter? Intercell membrane.
Intermembrane space.
There we go.
Right?
Yeah.
So just keep thinking, this is where, right? If you can remember, boom, right? That water falls. It turns a wheel, turns a turbine, electricity flows. Is the water part of electricity? No. Did those power plants use the energy in water to make electricity?
Yes.
Conceptually, this is what I can understand it. The cell is using the energy, here it is now, in the electrons, to increase what? So increase the concentration gradient of what, where? Joanna? Good, in concentration gradient of what? If I reverse yesterday's lesson, let's put NADH back, right? Let's say I haven't counted the protons in the intermembrane space. Let's just keep it simple. Let's say there's 100 in there, right? When this first reaction takes place, have we increased or decreased or kept the same amount of protons in the intermembrane space? We've increased it, right? Was energy required to increase the concentration gradient? What do we call that? Energy was used to move this proton from the matrix through the protein to the intermembrane space. That proton moved from, how would you describe the concentration of protons in the matrix compared to the intermembrane space? Low to high. When molecules flow from high to low, as we're going to see in a few minutes, that's diffusion. Diffusion is classified as what kind of transport?
Passive.
Passive, right? Can you tell me why diffusion is classified as passive transport?
Because it goes with the help of water.
Good, so water is colliding. Is the movement of these protons, if we're describing that movement using this word, is the cell, or specifically the mitochondria, using energy for that to happen? No. So, high to low, that's what we're going to see today. Protons, did we not talk about ET yesterday?
Oh yeah, we did.
Right, so for ET, in this case, the proton, I just made this up yesterday for the first time, it might not be perfect. For the proton to get home, the only gateway is through ATP synthase. No energy required for that. It takes energy to move the protons into the intermembrane space. We'll see the significance of that in a minute. Passive transport, what kind of transport is the movement of this proton?
Active transport.
Active transport. And why, what are the main concepts in active transport?
That it uses energy.
It uses energy, in this case the energy is in the form of what?
Electrons.
Electrons, good. And what else is connected to active transport? You got to talk to me about concentrations, differences.
Okay.
So what's the concentration difference?
Well, it's low to high.
Low to high. Okay, last example yesterday. Is this an example of you being in a canoe going downstream or paddling upstream?
Upstream.
You're using energy. Okay, we're going to see the benefits of the use of this energy today. Joanna, I know you're excited because you showed up today. I'm impressed. Okay? As I am with the other three students here. Okay, let's go. We're now going to talk about Q. So if I do this to represent, here's what's happening to Q in the electron transport chain. So this is why you will read, you read your textbook. So let's jot this down. Q is a shuttle. Okay, now squad. Now see what I'm doing here? I just have to move it off. Like Q is embedded in the phospholipid bilayer. It's just bouncing back and forth between. What's the protein that Q is now contacting?
NADH dehydrogenase.
Yeah. Good. And Victoria, what's the name of this protein? Okay, if you can't remember, that's fine. Just have your note out from the first day like we did yesterday. So you can see quickly what you've already labeled. So why don't you just take this out of your mind, your lip now. Is your hand okay? What's that? Okay. So what's the name? Where are we here?
BC.
Yeah, exactly. So Q is bopping between those two.
Okay.
All right. Speak to me like this, squad. Which protein do you think has a higher electronegativity? Q or NADH?
Q.
Q has a higher electronegativity. Each of these proteins in this amazingly designed system has a slightly higher electronegativity than the protein adjacent beside it. And we're talking about that. We're reading here in this direction, right, to your right. So check it out. Q here is going to collide with NADH. There's a pair of electrons here. Those electrons are going to get attracted to Q because why? Lauren?
Because it has a higher electronegativity.
Exactly. Q will then shuttle, boom, 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 like this. Shuttle the electrons to the BC complex. And what's the mechanism of the BC complex? Gaining those electrons. Clearly, my hand is moving means I'm going to show you a move later today where I'm actually getting magnetic molecules to bond together and I'm not physically going to touch them. Okay? Here, I am in the model moving the electrons. How's that happening for real in the mitochondria? James?
The BC complex has a higher electronegativity.
Exactly. Right? Okay, we good? Okay, now, squad. At this point, the electrons in the BC complex are virtually going to repeat what happened in the first enzyme, the NADH dehydrogenase. The electrons will move from one amino acid to another. What will that create?
Gaps.
That's right. So those proteins, here's an electron, right? When the electrons are on there, the proteins, BC complex will change shape. Gaps will form. And guess what's going to get moved from the matrix to the intermembrane space? Another what?
Proton.
Another proton. So in your picture here, have a proton in the BC complex is what?
Active.
Active site.
Oh, Mr. McClellan, I had a
Yeah.
question.
Yeah.
So, the electrons transfer because of electronegativity, but when they're in NADH dehydrogenase, they've created gaps and a proton's gotten through. So, are those things happening kind of in sequence? So, like the proton gets through NADH and then Q gets the electrons from the dehydrogenase?
Yeah. Well, yes. What you're not, I'm showing you an incredibly tiny picture of what's going on. Now, the picture you're seeing is accurate.
Yep.
What is not accurate is there's a massive amount of electrons in here.
Mm-hmm.
Right.
Okay.
Okay.
So there's just protons continually going through the dehydrogenase?
Yes.
Okay.
Yeah. But what you're learning here, when one pair of electrons enters, one proton gets pumped up. So if there's, say there's 50 pairs of electrons in here, 50 protons would get pumped up, right?
Mm-hmm.
But we're following, in this story, and we'll get really specific later, one pair of electrons from one coenzyme. Squad, I forgot to mention this. Where did this coenzyme get reduced? What part of the cell? Where did this coenzyme gain its electrons?
The matrix.
The matrix. So it could have been produced in the what, Joanna? Or the... Good. It was not. We're going to follow this tomorrow. It's not an NADH from glycolysis. Okay, excellent. So, check it out.
James, do you have white out?
You're going to move electrons with arrows through your protein.
Mm-hmm.
You're going to, that proton is going to get pumped up. I am now going to indicate, Victoria, you got mixed up, understandably so, with the number ones I had, remember? So why am I putting a number 2 there? That's right, it's the second proton that the energy, we reverse this, Victoria, look. Boom, 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 boom. These electrons were on NADH. One pair, boom, pumped up 1, 2 protons. So now the cell is increasing the concentration, right? We started with 100, now we have 102, correct? Yeah. Same pair of electrons. Okay, any questions? You're good. C is also a shuttle like Q. It's bopping to the B between the BC complex and the what? What's to my right here?
Oxidase complex?
The oxidase complex. James, what will C attract when it collides with the BC complex?
The electrons?
Because?
It has a higher electronegativity.
You guys are on a roll. Exactly. So now you can start to see a mechanism of the movement of the electrons. Hey, here's an interesting question. What's the name of these set of proteins we're studying?
The electron transfer chain.
The electron transport chain. Have you seen electrons transported?
Mm-hmm.
And they're kind of in a chain.
Like a school bus.
Pardon? School bus.
They're like a school bus.
Okay, so here we go. C, boom, boom. The electrons are now in what protein, Joanna? Atta girl, they're in the oxidase complex. I know this is repetitive. Victoria, what's the mechanism of those electrons being transferred from C to the oxidase complex?
The oxidase complex has higher electronegativity.
Good. Do you understand that word means what?
How strong it can attract electrons.
Exactly, that's right. What is the strength of the attraction for electrons? Very good. Okay, squad. Here, I want you to put, you're going to have an O2 molecule in the oxidase's active site and a proton in the active site.
Was it just one of the protons that was hanging out in the matrix?
Yes,
okay.
Matrix proton, correct. Now, when the electrons move through the oxidase complex, check it out. The same mechanism. what's going to happen to this protein?
It's gonna have gaps.
There's going to be a shape change, and that proton is going to be attracted to amino acids and electrons, and get pumped from the what? Matrix to the intermembrane space. Biologists are using the term pump because if you're pumping water by hand, you're using what? You're using energy. So this is energy. All right, squad, 3. All right. We good?
What does the oxygen do? Or are we getting to that?
Yeah, we're going to get to that. Rather significant. So therefore, we're now going to talk about why this is an aerobic, Lauren, why this is an aerobic system. So we'll get to that in a minute. Any questions so far? Okay, well, I will let you finish your note here for a second. Good. So let me check one thing here.
Well, this is going to be a bit I'm going to have to cut out.
Okay. All right, mini review. This is about the 25th time we've talked about this. Okay? Energy is in glucose in the form of what?
Electrons.
Electrons. Those electrons are transferred to
NADH.
and
FAB. Or FAB.
Right. Correct. Why are there so many enzymes conducting all these reactions? 8, 9, 10, 11, 12, 13, 14, 15, 16. Why so many steps?
Because they have different active sites?
They do have different active sites. Has to do with heat.
Oh.
Control the amount of heat release.
Control the amount of heat release one step at a time.
So they don't ignite.
So the whole thing does not ignite. Okay, correct. We're now following squad. Look, just one pair of electrons. We have, correct me if I'm wrong, we've increased the concentration of protons.
Yep.
If we went from my example from 100 to 103, I know that doesn't seem significant, but it is 3%, and we've only needed one pair of electrons, right? Any questions? All right, now you're going to make a note on aerobic / role of oxygen in the ETC. What's the role of oxygen in the ETC? Now you remember grade 11 bio,
Vaguely.
right? O2. Now, are we all breathing in oxygen right now?
Yes.
Do you know why? No. You know enough to know that if you don't have it, you die. So, and it's not that you just die, you die quick. Right? How long could you live without water?
3 days?
Yeah, right? Much longer period of time. This is clearly a critical molecule. So now you're going to find out the magic of this, really. So, this enzyme has an active site, boom, it splits O2 into single oxygen atoms. So that oxygen atom, you studied this in grade 11, or molecule I should say. You breathe in, goes into your lung, remember the alveoli? gets into your blood, heart pumps it around, enters a cell, diffuses into the mitochondria.
Do the electrons just kind of hang out there and protons keep going through?
No.
Okay.
Mr. McClellan.
Yes, sir.
Is Jacob Fonte there?
No, I think he's at the hockey game.
Oh, yes. Okay, thank you.
Thank you.
Hi.
Yeah. Okay, so here, what you're studying right now is think of this as the exit. How do the electrons get out of the electron transport chain? Right?
Mm-hmm.
I mean, if they didn't, a common analogy is, I mean, this is as you all have been on the 401 when there's an accident.
Yeah.
Sometimes you could be stuck on the 401 for like an hour because what gets backed up?
Cars.
So if the cars can't get off the 401, everybody's stuck in that traffic jam, right? Here, think of oxygen as the off-ramp. That's what it is. Anybody remember what the electronegativity of oxygen is? Now remember, when I say oxygen, we're talking about a single oxygen atom.
4.5?
3.5.
Oh,
so it's
yeah,
high.
it only
That's-
goes till 4.
Yeah. Yeah, so it has a high electronegativity, right? How many valence electrons are in oxygen, Joanna? 6, right? Okay. How many electrons can that one oxygen atom attract? 2. Do you think
I
that's
get
a
it.
coincidence? So, look, check it out. What I'm modeling right now is that oxygen atom attracts those electrons right out of the electron transport chain, specifically right out of the oxidase complex. So I'd like you to draw this almost step by step, right? Here's picture 1, right? Here's picture 1. Here's picture 2. Now in picture 2, do this squat. 2 protons. Draw arrows to the electrons in picture 2.
Oh.
Yeah, and what does that whole mean, word?
It means it makes water.
It makes water. Yeah, exactly. Who would have thought? So you have this picture here. Your third picture is this. You watch the flip move here. Boom. Boom. Right?
And this is happening with both waters? Or both oxygens?
Yes. So now, we're doing good. Who would have thought the electrons in the food we're eating, specifically the electrons in this story in glucose, end up becoming a water molecule? Who would have thought that the oxygen you're breathing ends up becoming a water molecule? Squad, this is about 15 to 20% of the water you and I need each day is made right here. No drinking, no eating, nothing, boom, water. Okay? Any questions?
I have a question, but it's like kind of unrelated.
You go.
If the ETC continuously makes water, why is it that people get dehydrated? Like, why do we run out of water if our body makes it?
Because if we're not taking water in,
You can't make more water.
right? So it's kind of like you're you're in the canoe and you're bailing the water out, but water's coming in at a faster rate. So yes, we're making water, but we're not taking enough in, and you get to a threshold point where you're losing more than you're producing. Any other questions? Okay, let's take a look at a couple things you know. So here's a couple of side notes. Let's just deal with this oxygen atom for a second. Let's just say that this single oxygen atom did not pick up a pair of electrons from the electron transfer chain. So here's the old double-edged sword, right? This is a beautiful molecule. Is there something commit or something? Hello? They don't know where we are. Did you guys hear somebody?
Yeah, it's a weird noise. It sounded like when you drag your magnets over the chalkboard, like that. Yeah. James, that's creepy.
Oh, no.
That's weird. What?
I know, but it's creepy.
Meiske.
Oh.
They're doing the slinky thing.
Oh.
Okay, so that's what it is. Okay, so this single oxygen atom has a high electronegativity. So I think the double-edged sword is this. One of the things, without being too morbid today, if you live long enough, this molecule can end up killing us slowly. Now we need it to live, and that's the trade-off. Use it, hey, use it for 90 years, but it's going to have a side effect. The side effect is, as an example, this oxygen atom could pull electrons off the phospholipid bilayer, damage it. We haven't started studying genetics yet. Pretend this is the nucleus. Could go into the nucleus, grab electrons off of DNA, cause mutations.
That's not good.
Not good. Do you know what they call this? This is a free radical. Have you heard of free radicals? Okay, we're going to do a lab on some enzymes. I'd like you to show you something that you're familiar with. Look at this squat. Now, I'm just changing my model here for a minute.
Mm-hmm.
This is not carbon, that's representing vitamin C.
Okay.
Vitamin C is kind of, you can think of it like a coenzyme. It carries extra electrons.
Mm-hmm.
If it comes into contact with a free radical, in this case oxygen, look, it can pass a pair of electrons to oxygen. What will that oxygen now attract?
Protons.
Protons, and produce what?
Water.
Water. Are you familiar with this language? Vitamin C is an antioxidant.
Oh.
Right? So do you
Yep.
see that connection? So vitamin C, which we all need, hangs out in the water part of your cells. In this case, the water part of the mitochondria. So that means vitamin C is water soluble.
Is that why when people used to get scurvy, because they didn't have enough vitamin C, they'd get dehydrated?
One of the things, yes. And damage would occur to cells. Now, some people are doing experiments where they're eating massive amounts of vitamin C. Right?
Can't that kill you?
Well, the data so far is no. Because what happens is the extra vitamin C is in the water part of the cell. So it goes to the kidney, goes into urine, you urinate it out. Vitamin E hangs out in the lipids, hangs out in the phospholipid bilayers. So vitamin E can build up and stay in the body and become toxic if there's too much of it.
Mm-hmm.
But vitamin E does the same thing, except it's working right in the phospholipid bilayers. Okay, good. Excellent. Let's get to the magic. Now, squad, the concentration gradient. When we say gradient, what does the word gradient mean?
Direction.
The direction. Why is the direction of movement of protons going to be towards and through ATP synthase?
Because the protons need to get back home.
They do. And what is the only protein they can diffuse through?
The ATP synthase.
Yeah. So if we jammed up this classroom with, I don't know what, 200 students, and everyone's pushing everybody,
Going that way.
and I open that door, that's where they're going to fall out, right? Okay, good. So let's jot this down. One proton, and let's be clear here, squad. I'm not talking specifically about the protons that were just pumped up. What's happened now is the concentration of protons is staying at a high level so that a proton will diffuse through We're going to place it right here through ATP synthase.
Okay, so the high concentration of the protons in the intermembrane space forces other protons
To
through the
ATP.
ATP.
Exactly. You hit the nail on the head. As opposed to your thumb. All right, now, let's check this out. This protein is more like this. Look at this, I built this in October 2005. Okay, see it's like this? So it's got 3 sections to it. Here's the phosphate and here's ADP. I'm going to show you this in a different way in a minute. But when that proton squad moves through the protein, is there a charge on the proton?
Yep.
Yes.
Yep, you're not convinced. What is a proton? You studied this, Joanna, way back in grade 9. Where do you find protons, young lady? On atoms. Where in atoms? Good. And what charge do they have? Exactly. So hydrogen, when it loses the electron, all that's left is a proton.